Good afternoon. Welcome. I'd like to introduce Chris Elias, the president of the Global Development Program at the Bill and Melinda Gates Foundation, to lead this very timely panel on the fight against Ebola.
Thanks, Jeremy. And thank you all for being here this afternoon. And thank you for joining us for this panel discussion on accelerating interventions needed to fight the Ebola epidemic. I'm delighted to share the stage this afternoon with this esteemed panel of leaders in the Ebola outbreak response. And thank you for taking your time, panelists, for sharing your insights with us today. The 2014 Grand Challenges meeting presents a particularly timely opportunity for us as partners to explore how the global scientific community can work together to fast-track solutions and deliver them on the front lines during this period of crisis, especially now, given the situation we're also actively tracking and working to improve in West Africa. As we discuss the current status of the Ebola outbreak in West Africa, the goal of this panel today is to explore the unmet need for new Ebola interventions, namely vaccines, drugs, and diagnostics, and the role that health system strengthening can play in this solution. Before I begin, I'd like to just simply acknowledge all of those working across many institutions on the front lines of the Ebola response, and to thank them for their tireless efforts in these challenging and dangerous conditions. I'll start by sharing some background on the current epidemic and where the Gates Foundation, alongside many other global partners, is contributing to the outbreak response. But I'd like to start by introducing our panel, and then I'll call on them in this order. First, we'll be joined by Vasi Satyamurthy from the World Health Organization, who's joining us from the World Health Organization in Geneva by video conference, and we'll have him speak first because it's quite late, and we appreciate Vasi staying up late to be with us this evening. Vasi works in the Immunization, Vaccines, and Biologicals Department for the WHO, which focuses on vaccine-preventable diseases, vaccines, immunization, and immunization policy worldwide. Following Vasi, we'll hear from Neil Ferguson. Neil's the founding director of the UK Medical Research Council Centre for Outbreak Analysis and Modelling, and he's also head of the Department of Infectious Disease Epidemiology at the Imperial College of London. His work focuses on using mathematical and statistical models to investigate the processes shaping infectious disease pathogenesis, evolution, and transmission. Neil's been working closely with the World Health Organization on the epidemiological modeling of this outbreak, and we're delighted to have him with us today. Following Neil, we'll hear from Francis Collins, the director of the National Institutes of Health. Francis oversees the work of the largest supporter of biomedical research in the world, spanning the spectrum from basic to clinical research. Francis is also joining the final plenary panel later this afternoon, so we'd like to thank him for his willingness to be on stage all afternoon. Then we'll hear from Ted Bianco, the director of innovations at the Wellcome Trust. I've known Ted for many years, and I'm glad he could join us today. Ted founded the Division of Technology Transfer at Wellcome in 2003 with the primary goal of translating basic research into health products and public health tools in areas of unmet medical need. Then we'll hear from David Vaughan, the head of external research and development in North America for GlaxoSmithKline. David works in vaccine and pharmaceutical R&D to combat infectious diseases worldwide. I'm looking forward to hearing his insights on the role that GSK is playing, particularly in accelerating vaccine development. And last, but certainly not least, I'll be pleased to introduce Chris Karp, the Deputy Director of Global Health Discovery and Translational Sciences team at the Bill and Melinda Gates Foundation. Chris leads the Vaccines and Host Pathogen Biology team that targets fundamental scientific and technological advances in global health that could lead to new ways to prevent and treat disease. If I can have my first slide, just in case anyone didn't actually, next slide, the map, there we go. In case you haven't been reading the papers, it's been hard to not read the papers given the pace and the scope of this epidemic. This is the first outbreak of Ebola in this part of West Africa, and it's the largest outbreak that we've ever seen. And this gives you a snapshot. There are 3 heavily affected countries in Guinea, Sierra Leone, and Liberia. The overall case fatality rates in those countries are between 50 and 70%. And there have been some spread from those countries to 2 other countries in West Africa, Senegal and Nigeria, as well as the transmission of a case in a Spanish hospital. Neil's going to tell us in some more detail about some of the modeling and the patterns of, in the 3 heavily affected countries. I would just try to pull out a positive story amidst an otherwise dim set of news stories about Ebola, and that is the efforts of the government of Nigeria in containing the outbreak that started there almost 2 months ago. There was a patient from Liberia who traveled to Lagos, got sick on the plane, was brought to a hospital. They had an outbreak, but partly because of rapid action. I think this teaches us some important lessons for protecting other countries in the region and around the world. Rapid action, in this case, using some of the assets of the emergency operations centers that the government of Nigeria has established for polio eradication. People and equipment, et cetera, were mobilized very quickly with a very effective contact tracing, et cetera. And while there were some cases that spread from Lagos to Port Harcourt, the total case number from that introduction was only 20. And now they're out beyond the incubation period and have said confidently that that introduction's been contained, although they're obviously significantly at risk of other introductions given the transit point that Nigeria is. The other thing I'd like to do by way of introduction before turning to Vasi is to go to the next slide, which just shows, I think all of you probably heard in early September that when we saw the pace of the Ebola outbreak picking up, Bill and Melinda generously agreed to contribute an additional $50 million to the global outbreak response for Ebola. And there's been a lot of questions over the last couple of days about how we're spending those funds, so let me just give you a quick rundown of that. We basically are using that $50 million of emergency response in 4 pillars. The first one, which we spent actually in the first week that after the funds were approved, was to fuel the global response. One of the things we heard from our key partners at WHO, UNICEF, the CDC, was the need for flexible and immediate funding. So we provided all 3 of those organizations with resources so they could get going while larger sources of funding were coming and being appropriated, et cetera. The second pillar is research and development, and that'll be the focus of our panel today, where we're looking, as you'll hear from Chris, at a range of different contributions alongside groups like NIH and Wellcome for vaccines, therapeutics, other countermeasures, as well as diagnostics. Our third pillar has been country-level response, and here we've worked particularly closely with the CDC, as well as the governments of the affected countries. So we've been very active both in Nigeria and Senegal in strengthening the emergency operations centers to both help contain their introductions as well as prevent and prepare for potential future introductions in rapid response. And we're working with partners, including CDC, in countries in Liberia and Sierra Leone as well. And then our fourth pillar is emergency preparedness, where we're looking at how we can better strengthen the firewall, if you will, in high-risk countries that are currently unaffected, looking carefully at the modeling data that Neil and other modeling groups are producing to help understand where the vulnerability risk map is in West Africa so that we can strengthen preparedness and emergency response when and if that happens. And that's how we're spending that $50 million of extraordinary funding that Bill and Melinda provided for this acute outbreak. Importantly, however, on this slide is Pillar 5, which is that we have begun to think across all of our strategies, if you will, the bread and butter of the Bill and Melinda Gates Foundation, our agricultural development, nutrition, primary health care, et cetera. How do we think about helping this region recover, and how do we think about building resilience? Because part of what the Ebola outbreak has exploited is the weak health systems in these post-conflict states that have left these communities and countries so vulnerable. So that's what we're doing in terms of our response. So with that, let me turn to Vasi, who I think is on Skype. and asked Vasi to share with us an update from the WHO about what they see as the highest priorities for new interventions to aid in the fight against the Ebola epidemic, and to hear more about what they're doing. Vasi, welcome.
Thank you, Chris. Can you hear me okay?
Yes.
Great. So, first of all, it's a great honor to be on this panel, if only virtually. I apologize I can't be there in person. Ironically, it's the Ebola crisis that is preventing me from being there in person. We're working 18 hours a day, 7 days a week to do exactly what is the topic of this panel, fast-track Ebola interventions. And I'm going to focus on the Ebola vaccine situation. I am responsible for the acceleration of phase 1 Ebola vaccine testing at WHO. So if I could just share with you where we were when we really started to move into the most aggressive phase of acceleration in August. We were faced with multiple manufacturers we needed to prioritize. I think from the vaccine side, the prioritization is in a sense more straightforward than the therapy side, in that there are two vaccines with which satisfy two simple criteria. So there are two vaccines with really strongly supported preclinical efficacy data, as well as GMP-grade clinical material. And we have been public about this. We're clearly neutral when it comes to which manufacturers we'll work with, but those are the two criteria that we chose. And we have worked, and I think this will be seen as a case study, and I'm sure that everything that we're all doing in this time of crisis will be analyzed, already being reported in the media pretty much in real time. You will have seen that, as one example, the meeting that we had in Geneva last week was being reported as the discussions were being had. So it's good to think about, are we doing everything we possibly can? And I think in many ways, this is best practice for accelerating evaluation of experimental interventions. There are a couple of ways that we've been able, and by we here I mean all of the partners involved, so NIH, different parts of NIID, multiple other funders, and the global scientific and public health, and in my case study in particular, the clinical trials community has come together in ways that I have never seen before. And I've heard this again and again, talking to many people that have been involved. Nothing can delay the timelines. Everyone is working together to move the timelines forward. So just to give you one example, we have been working to accelerate the phase 1 timelines. We were in favor of phase 1 African data, in addition to phase 1 data from Europe and North America being generated to high quality as quickly as possible. We understand that the first African Ebola vaccine data should be emerging later this week, if not later this week, then next week from Mali. And that has taken about 7 weeks from the first phone call from myself to Professor Samba Sow in August to the first vaccination in Africa with a novel phase 1 vaccine trial. Now, I personally am not aware of those timelines having been met before. And we're aiming for generation of safety and efficacy data in potentially a 9-month time frame. We're trying to go even faster than that. I mean, nothing is fast enough. When you see what is happening on the ground in Guinea, Sierra Leone, and Liberia. But I think by everybody working together and really looking at all of the timelines and problem solving pragmatically to try and drive those timelines forward, we have been able to take what may have been a 4 to 5 year timeline and move to, I think, what in the most optimistic scenario could be a 7-month timeline from the very first phase 1 trial to a safety and efficacy database. And some of the really critically important partners here have been the two manufacturers, so GSK in collaboration with NIAID, the company in Newlink have worked closely with the Canadian Public Health Agency, the Public Health Agency of Canada, and as many other partners. And a couple of other points that I wanted to make. Generally, in order to get clinical trials done quickly, there are at least 4 major constituencies. So there's the regulators, the ethics committees, the manufacturers, and the funders. And all of those have stepped up to the plate. A really important part of this is flexible funding, as you talked about, Chris, and the Gates Foundation has played a major role, as has NIH, as has Wellcome Trust, together with others. And we've seen funding that's been agreed in days rather than months. So that's been absolutely critical. The other important point is that none of the quality assessments, none of the scientific rigor can be compromised, and it has not been. But what has happened is that the timelines have been moved forward by the authorities that are responsible for the approval processes, doing the same high quality review in greatly accelerated timelines. So the US FDA has been phenomenal in the support that they provided. The MHRA in the UK, as another example, went through the whole process in 4 working days, despite the fact this was the first in human testing of that particular monovalent chimpanzee adenovirus product. The ethics committees have been phenomenal, and the clinical trialists have been incredible. So it's all going as fast as it can. Would we like it to go faster? Yes. Is there anything else that we can do right now to make it move faster? No, there isn't. I think another part of this is the parallel pathways. So we set this up pragmatically, working with the network of partners here to have some kind of semi-redundant steps. You would normally have sequential steps in clinical development pathways. Here we have parallel steps. So we have the USA and the UK trial essentially happening in parallel. We have 2 African sites identified so that if there's an unforeseen hitch at any one of the African sites, we could move to the other one. And I think that's all been very helpful. And then I'll end just with a couple of minutes talking about the recent meeting in Geneva last week. So I think that this is a good example of, meetings in Geneva don't necessarily seem like the most exciting thing to talk about, and in general they aren't. But in this case, this was a good example of where we came out with something that I don't think anybody necessarily saw we would necessarily come out with, and we could only do that by having really very strong representation from the affected countries. We had clinical researchers, clinical trialists, those who had been involved in treating on the front line in Guinea, Sierra Leone, and Liberia, as well as many from the neighboring countries which have been providing support, Ghana, Mali, Senegal, just to give some examples, Cameroon and Nigeria, as well as regulators of the US FDA, EMA, many of the African regulators, ethicists, clinical trialists, and a lot of the best methodologists in the world were there. And what we came out with from the meeting was clearly not everybody in the room agreed on everything. That never happens, but there was a clear sense that the fastest way to generate safety and efficacy data in the context of this emergency is for parallel tracks with phase 2, what are being called phase 2A studies in neighboring countries, which is the best way to rapidly generate a robust safety database using clinical trial sites with a track record for vaccine trial data to regulatory standards. And in addition, in parallel, not waiting for those studies, but in parallel, moving to determine efficacy in affected countries in populations at the highest risk pre-exposure. And we moved from only talking about healthcare workers as that group to frontline workers during this meeting. And one of the things that I think came out from the meeting was that it is clear that scientifically the most desirable approach is randomized control trials. Ethically, it is also clear that we are in equipoise now. We do not know if these vaccines work. We have supported preclinical data, but we are in equipoise. So it is ethically appropriate to conduct randomized control trials. And this was clear at the meeting. Now, does every single person in the world actually agree about that? No, but at the meeting, this was agreed. Now, the next step is clearly the authorities and the decision makers at the sites in the affected countries are going to be critical in the finalization of these trials. Those discussions are already starting. And if they feel that it's not feasible or practical to conduct randomized control trials, then we need another option. And so we would encourage that the manufacturers develop both options, so randomized control trial options and an alternative option. And there was a lot of discussion about stepped wedge designs. I saw interesting attempts by journalists to explain what a stepped wedge design in various media outlets. But there clearly are several alternatives to the traditional randomized control trial. I think both of those need to be considered, but the RCT design was considered to be scientifically highly preferable and ethically appropriate. So perhaps I can leave it there, and I'm happy to take questions later. I'll hand over so that there's time for the other panelists. Thank you, Chris.
Thanks, Vasi. Neil, can you... Neil, can you share with us an update on the epidemiologic modeling and what it's telling us about the course of the outbreak and what tools are most needed?
Yeah, absolutely. I think it's useful to put the novel intervention development effort into context against the wider control effort in place to control this epidemic. And I think it's informative to think about why control has failed thus far. First of all, Chris mentioned in his introductory remarks, briefly, health systems. And we're talking about, with Liberia and Sierra Leone, countries with almost the lowest number of doctors per capita in the world, with no really effective infectious disease surveillance. And so whilst Guinea started getting on top of this outbreak within several months of it starting in that country, we had circulation of the virus really unchecked for 2 to 3 months in May and June into July in Sierra Leone and Liberia. What we're facing at the moment is an epidemic which, through enormous efforts, seems to be plateauing off in Guinea, certainly, and there's some evidence of a plateauing in cases in Liberia. It's too early really to be certain whether this is a real plateauing or frankly reflects underreporting of cases and delays in data capture. In Sierra Leone, I think the situation is still much worse. There's no evidence of a slowing of the epidemic there. And really, certainly in both Liberia and Sierra Leone at the current time, the number one problem is what it's been for the last few weeks, is a shortage of trained staff to conduct contact tracing and hospital beds and isolation units to isolate suspect cases. And so we have now 8,400 cases as of today, and that number is going to continue to rise substantially. Now, in the short term, the efforts to control this outbreak have to be the traditional efforts we know work against Ebola. And that means putting more Ebola treatment units in on the ground, training more staff as rapidly as possible. The one novel intervention which could make an enormous difference in the next 1 to 2 months, if it was available, and I realize that's a big if, is more rapid diagnostic testing. A substantial portion of the beds in the Ebola treatment units are being occupied by people who, at the moment, are either not being tested at all because of lack of diagnostic capacity, or who are tested and in many cases test negative. And those beds could be used by the people currently who are queuing up or not able to get into a hospital bed who truly have Ebola. The key problem we face, and the reason the epidemic is growing, is that we're not getting people with Ebola out of the community fast enough and stopping transmission in the community fast enough. So that's the broad context. Do I think vaccine is going to play a key role in controlling this outbreak? I do hope not. If we're in the position where vaccine is required to stop transmission, then it's going to be a very, very bad situation because we're not going to have substantial amounts of vaccine sufficient to affect transmission until the end of this year. I very much hope the role of vaccine will be to protect as soon as possible people at particularly high risk, and potentially longer term to protect these vulnerable larger scale populations against future repeats of this catastrophic outbreak. I think I'll call a halt there.
Thanks, Neil. Francis, I know you've been spending a lot of time on Ebola. Want to tell us how it looks from NIH?
Well, indeed, there are almost daily phone calls involving my boss, Secretary of Health and Human Services, and we're working closely with the Department of Defense, who, as you know, is now trying to put in place 1,700 beds in Liberia as quickly as possible. And obviously with USAID, who made an announcement yesterday about a grand challenge, which I hope you all paid attention to, especially in the area of needing to have better kinds of personal protective equipment. From NIH's perspective, our closest colleagues are at CDC, and they are very much in the front lines trying to manage the effort both in West Africa and, of course, now with a great deal of focus on what's happening in the United States, particularly in Dallas. And you may have heard there's an announcement today about doing additional screening of passengers coming from the affected areas, not just exit screening, but also entrance screening, at least beginning at JFK Airport on Saturday. From NIH's perspective, our role very much is in trying to develop and as quickly as possible test in clinical trials, both vaccines and therapeutics. We've been working on vaccines actually since 2001 for Ebola. The current vaccine, the chimp adeno 3 that was briefly mentioned by Vasi, is a 5th generation effort and one which looks very good in the non-human primate model, but which until very recently had not been into humans. Again, much credit to our colleagues at the FDA who really pulled out all of the stops in order to get through the approval process. We have, in fact, now run a phase 1 trial with healthy volunteers at the NIH Clinical Center on that chimp adeno 3 vaccine, which is an important partnership with GSK. And those 20 individuals have not had red flags, so at least we are not concerned that there's going to be a frequent problem in terms of safety. We will not know, however, what kind of immune response is possible for another month, so we wait for those results. Meanwhile, we have a collaboration with the Wellcome Trust, and Ted will talk about that, which enables testing of a monovalent version of that same vaccine. The one we're testing at NIH is a divalent, to see how that works in other settings. In addition, as you know, and as was mentioned by Vasi, there is this VSV vaccine developed by Public Health Agency of Canada and being produced by Newlink, which is close on the heels of the chimp adeno 3 and will begin trial testing in a couple of settings as early as next week to see also how it performs in a phase 1 trial. Obviously, the big question is, if it looks as if you have safety and some evidence of immune response, does this really work in the real world? And we strongly agree with the conclusions that Vasi mentioned of the recent WHO meeting, that you cannot assume that. And we are at equipoise. And we have to remember that there can actually be negative consequences of distributing a vaccine without having evidence of efficacy. For instance, people who may assume that they're protected and relax their own care as far as exposures. Or just let's remember, there was that HIV vaccine that actually increased infectiousness to those who received it. There are possibilities there, hopefully not commonly encountered, but not unheard of, that the vaccine could actually turn out to be harmful and having a reverse effect on efficacy. We just have to know the answer to that. There is a meeting tomorrow and the next day in Monrovia with CDC and NIH and Liberian government officials to figure out what would be the appropriate way to begin to initiate the design of a randomized control trial, which would have a control arm. In my mind, it would even be better if we could synchronize all of this, that we might have a 3-arm trial, which was basically a control, the chimp adeno 3 and the VSV, all basically done head-to-head, and then we could do the maximum in the time available and have a single control arm instead of having to have 2 separate trials. Whether that's going to be possible or not is not obviously something that's easily determined. Again, I think all hands are on deck here in terms of pushing this forward. Let me just say a word about therapeutics, another area of intense need. I think all of you are familiar with ZMapp, the cocktail of 3 monoclonal antibodies being produced in tobacco plants, which was available in only very small amounts for those 7 patients who received at least a partial course of this, who were quite ill with the disease, and 5 of whom survived. But obviously, that's way too small an N to draw any conclusions about whether that therapeutic contributed to their survival or not. This week's Nature magazine reports the results of that ZMapp in the non-human primate model, making it clear that it works really well, and you can even wait until monkeys are 5 days into their illness and receive a very good response in terms of protecting against death. So this is a very promising but unfortunately frustrating situation because of the very complete lack of availability of the kind of GMP material that we wish we had right now in order to do a trial in a more controlled way. Producing this in tobacco is something that we and the Gates Foundation and others are trying to see, is there a way to accelerate? But frankly, that is not a trivial process. And many of us are really looking harder at whether this could be shifted into a CHO preparation. There are all kinds of technical reasons why that's not quite as straightforward as you might think, because one of those 3 antibodies seems, at least in initial testing, not to be as potent in CHO production as it is in the tobacco plant, which is why they went to tobacco in the first place. But believe me, every effort being made to try to figure out a way around that, because we desperately need those therapeutics now. There are other therapeutics out there that have not been in clinical trials as yet. The BioCryst small molecule, an adenosine analog. There are siRNA preparations from Tecmira. There is one compound which you may have read about in the last little bit, which is actually something NIH had invested in over some time, which has actually been given now to 2 infected patients. And this is this small molecule called brincidofovir, which is basically orally available, has already been in 1,000 patients for treatment of adenovirus or CMV, but appears to have activity against Ebola, at least in vitro. And because of the fact that it's further along as far as human use is also looking attractive, but again, I would argue, has to be subjected to a rigorous trial before we should draw any conclusions about whether this is going to be the solution. It is appealing because there are a lot of doses available, and potentially, if this does turn out to work, this is closer to the kind of outcome that we all desperately seek to see happen. But again, we got to find out whether it works or not, and we got to go well beyond some in vitro study. Finally, I would say in the diagnostic arena, I agree with what Neil said. We desperately need to be able to get quick answers to whether somebody is infected or not. Most of the tests are currently PCR-based. Lots of people working on ways to speed those up. There are some antigen-based assays. We've been supporting one at CoreGenix. The question is, are they sensitive enough to be able to say with confidence whether somebody's infected or not? That kind of testing still remains to be done. So huge amount of activity, and again, I'm honored to be on the panel with all of these folks, all of whom are partners in this effort, whether it's Wellcome Trust or GSK, and Gates Foundation, Oxford, countries in Africa. This is the greatest possible argument to work together when we see these hundreds of people dropping every week, and the numbers are truly staggering when you look at that projection that's going forward. We have to figure out a way to stop this terrible outbreak.
Thank you, Francis. Ted, let's get your view from the Wellcome Trust.
Thank you. Within about 6 months of Jeremy Ferrara, new director, arriving in post, this broke. And coming from 18 years in Southeast Asia, seeing chicken flus and SARS and other viruses swim by, he probably thinks out of the frying pan into the fire and has been flat out, as many of us have. Wellcome's put out 2 calls for proposals in the month of September. And one of those is around clinical tools, the other a joint call with the Department for International Development in UK for an emergency response to humanitarian health crisis. Dealing with the first, there has been a very rapid movement, as we've heard, to get the chimp vector into phase 1 and ultimately immunogenicity testing. Wellcome, working with the Jenner Institute, Adrian Hill and his colleagues, and with GSK, has taken a monovalent construct, and as of 17th of September, there was the first dosing of a subject. As of current time, 15 subjects dosed with 3 doses and permission to instigate phase 1 in Mali. Where we are over the Canadian VSV construct is that we want to facilitate some of the coordination necessary between the parties to facilitate those trials. We have made agreements very earlier today in London to support work to look at whole blood and plasma transfers when the facility to achieve that is in place. And we have agreed to fund Peter Horby working with WHO, ICERIC, to establish a clinical platform which will be located in sites within West Africa where one can deliver highest achievable standard of care while evaluating various interventions. Peter will be visiting with MSF sites next week in West Africa. In terms of the Ebola outbreak, I think we're all very aware this is way more than an infectious disease outbreak. This is a humanitarian, sociological issue of great proportion. So the joint call with DFID has taken somewhat a different stance. Here we're looking at shortlisted bids now. There was a response of 200 bids within a matter of days. Those have been shortlisted and decisions will be made within the next fortnight. Things in that shortlist are dealing with modelling so that and projections so that one can use the best resource allocation and planning processes, trying to understand what needs to build trust in the community, why presentation for treatment is considered at the moment to be an issue, a very significant issue, what that would mean in the future when we have new tools to introduce into the area. There's a real necessity to have built confidence to understand what the West African general population and the healthcare providers need in order to garner that trust, to get the coverage that ultimately will be needed to bring the epidemic under some sort of control. So we are also very aware of additional tools like diagnostics to aid in case detection, but case tracing and the fate of survivors is also a very important feature. So there's a range of proposals that we're looking at which are really to do with less the medicine, more the sociology of this outbreak.
Thank you, Ted. David, can we turn to you for the insights from GSK and the private sector view on some of the vaccine challenges?
Certainly. I think we've heard already that vaccines under development represent a potentially effective intervention. In the case of the vaccine developed by the VRC and being taken forward into phase 2 by GSK, the data look very good in monkeys. But as we know, mice lie and monkeys don't always tell the truth. So we won't know that the vaccine works in people until we have the data that shows that the vaccine indeed does work. There is good reason for confidence, optimism in this particular vaccine. We've heard about the extensive work done at the NIH since 2001. This particular vaccine has been under development for more than 3 years by the Vaccine Research Center and working with a Swiss-Italian company called Ocairis. GSK acquired that company in May of 2013, and that's how GSK got into the Ebola vaccine business. The vaccine itself is a live recombinant, replication deficient adenovirus that vectors Ebola virus surface protein, glycoprotein, to the vaccine recipient. We're looking for a good antibody response, but using a live approach in the hope that we'll see robust cellular responses as well, particularly CD8 responses. This particular adenovirus serves as a vector. Was isolated from a chimpanzee in Louisiana with a cold, and it's designated chimpanzee adenovirus subtype 3. In terms of other reasons for optimism, similar vaccines have been in people, particularly chimp ad-vectored hepatitis C virus vaccines and malaria vaccines in more than 1,000 people. We've heard about the phase 1 trials. 3 have started in the US, the UK, and in Mali, just today, I believe. There may be as many as 9 phase 1 trials that will start this year. It's a huge effort, a lot going on in parallel. This will exhaust the available vaccine that has been generously provided by the VRC to all of these partners. GSK has started production of new lots of vaccine. The first new lot will be funded by the Wellcome Trust, and the second lot by the Bill and Melinda Gates Foundation. We hope to have additional vaccine available by the end of the year. So in terms of next steps for clinical development, we've heard from Vasi about the discussions in Geneva at the beginning of last week, and GSK intends to largely follow that path. We're targeting to start phase 2 testing in January of 2015 in more than 3,000 individuals, including children. This will be conducted in established clinical research centers in Western Africa, but not in the outbreak countries and surrounding countries. So this is an opportunity to generate additional high-quality data that will inform us as to safety and immunogenicity. So what we've talked about so far is fairly routine approach to clinical development, although greatly accelerated and many things happening in parallel. This possible because of the tremendous cooperation between many parties, and regulatory authorities have been mentioned in particular. What may be a little bit different that's been mentioned already is that GSK proposes to start a phase 3 randomized, controlled efficacy trial in parallel to the phase 2 study. So we hope to start that in January. This would be in frontline healthcare workers at greatest risk. And we would work to identify as quickly as possible and as completely as possible Ebola virus disease cases so that we can have an interim analysis, and if the vaccine is shown to work effectively, we can have immediate crossover vaccination to control recipients. But as already has been mentioned, the null hypothesis for a phase 3 trial is that the vaccine does not work. And in fact, we use two-sided calculations for the statistics, not ruling out that the vaccine could make things worse, increase the risk of infection or increase the severity of disease. So we'll want to get that information as quickly as we can. Now, all of this will be extremely challenging. GSK is still assessing the feasibility with the WHO, with government agencies, with regulators, with other partners. And we still need to come to a common understanding as to what is the best path to get us there with an effective vaccine that can make a difference.
Thank you, David. Chris, as I mentioned in my introduction, research and development is a key pillar of our part of our Ebola response at the Gates Foundation. Maybe you could tell us a little bit about some of the work that your team's doing.
Sure. So in general, to delve into your pillar 2, we're thinking as a foundation really in 2 tranches. The bulk of the work that's being done is aimed at things that can get out in the next 6 to 9 months. So along the lines of what Vasi mentioned early on, things for which there is a strong preclinical proof of concept, as well as the ability to get into humans quickly. So our work on that score has largely been mentioned already. So it's helping partners with accelerating production. So with GSK, with ZMAP, with moving that cocktail into CHO cells, et cetera. Additionally, there's work that's being done on diagnostics, both diagnostics for triage as well as for contact tracing and for reference labs. There's also an initiative, and I'd have to say that we don't actually have good preclinical proof of concept on this, but it's an approach that has been used in many different infections in the past, and that is immune globulin. So there's an initiative looking at both the feasibility and the practicalities of generating and then testing immune globulin. And so that's where the bulk of the effort is at the moment in that first 6 to 9 months. But while I share Neil's concern that interventions past that may in some way be failure strategies for the current epidemic, but certainly necessary for future ones. We are doing work aimed in the more 18 to 24 month time frame. So there is clinical preclinical proof of concept for antibodies. I think we all think that better antibodies are likely to be needed, both higher affinity antibodies as well as given the drift and the immune pressure the virus is now under, cross-reactive antibodies that can work both in this and future outbreaks. And so one thing that has been put together is an initiative to isolate and characterize and down-select better human monoclonal antibodies. And this has been a very gratifying initiative with a variety of partners, including the Wellcome and different US government agencies, biotech companies, and academic investigators, trying to take a variety of best-in-class, both complementary and orthogonal approaches, both isolating antibodies from survivors using classical techniques, as well as taking advantage of novel technologies that can do very high throughput, rapid antibody identification and characterization, as well as using tools like the humanized immunoglobulin mouse, which provides another orthogonal way for going at this. And I would say that it's been gratifying because in this time of emergency especially, the non-technical barriers, and I think the technical barriers are actually fairly low to identification of good antibodies, but the non-technical ones that you worry about in terms of barriers of ego and turf just aren't there, and that's quite gratifying, I'd have to say. That said, there are other barriers of perception and politics and bureaucracy in terms of, for example, transportation and collaborative access and getting critical samples necessary for this. And I think that's something that'll have to be worked on both sensitively and rapidly. So.
Thank you, Chris. And thank you to all the panelists. We have about 17 minutes left. We're going to open it up to questions from the floor. There's microphones on either side. Make your way up. Remember to introduce yourself, and if you want to direct your question to an individual, do that, or to the whole panel. And maybe while people are coming up to the panel, maybe I'll ask Francis a question, which is, we've been hearing a lot on the news that, while it's promising the kind of collaboration that's been described by many and the rapid attention to accelerating interventions, that we may have been less prepared than we could have been had we not had the significant budget cuts that we've seen at NIH and CDC and WHO and others. Do you want to comment on that?
Well, I'm afraid it's true. When you consider this has been a 12, 13-year effort in terms of getting the vaccine ready for what we all hoped wouldn't happen, but which now has. Clearly, we could have gone faster at NIH had we not been through what's been a pretty tough 10-year period, ultimately now losing about 23% of our purchasing power for research, which means everything has been slowed down over what it could have been. This is an example, but there are many others. In the vaccine arena, we're not as far along as I think we could be with a universal influenza vaccine or even an HIV vaccine. And again, we're working with wonderful partnerships in those areas, especially including the Gates Foundation. But it's a concrete manifestation of the circumstances that have afflicted the support of American biomedical research, at least through NIH, over the last 10 years. And I don't think that's being overly dramatic. That's just the facts of the matter. I'm sure our colleagues at WHO are suffering in a similar way by having really serious financial challenges in the midst of what is a very pressing global emergency. And it just comes down to that. If one wants to be prepared, it costs money to do so. We have been shortchanging that over the course of the last decade, at least in the US.
Thank you, Francis. Maybe we'll start on this side.
Matthew Albert and Sue Pasteur. I was curious to know whether or not there's been consideration about a prime boost strategy. Prior adeno-based vaccinations have required 2 different viruses, and it seems like there's a possibility for a 4th arm using both the adeno and the VSV.
So if my colleague Tony Fauci was here, he would answer that question like this. And again, I should have said at the beginning, almost everything I'm telling you about vaccines is information that I've learned from Tony and from Cliff Lane, who are in the middle of this minute by minute, hour by hour. It is the case, apparently, that the chimp adeno 3 vaccine does not have a long-term immune response. At least that's my understanding. You might need a boost if you wish to have a protection going on many months. But in the acute phase, in the monkey model, it's not necessary. David may have more data to offer there.
Yeah, I would refer you to the paper by Daphne Stanley et al. From the VRC. There's a number of non-human primate work that is reported there, but one of the most important is to use chimp ad as a single dose prime, and then at 8 weeks they give an MVA, Ebola virus boost, and they saw complete protection in cynos 10 months after that boost. So that's the first. But even with a single dose of chimp adeno vaccine, they've seen 50% protection 10 months out, which is pretty good. I mentioned mice and monkeys not always telling the truth, and I don't know what the translation will be from non-human primates to human primates. In the cynos, there's a fairly quick disease period, 6 days, but the incubation period is longer in humans, then a longer illness period. Maybe there's more time to build on that boost or that prime that's provided by the vaccine. So maybe we can have some optimism for even better, longer protection with vaccine in people. We'll see.
Ted?
Yeah, I mean, I think it's also going to be a massive logistical problem to prepare 2 constructs and to get them into people in the right order in an infrastructure which is struggling to cope with any kind of intervention. So although I can imagine highly desirable, it's really the practicality that would concern me.
Can I add something? Just to support the comments, and I think scientifically there's really a lot of information that would point one to indicate that an MVA boost may well be helpful, not only from Ebola, but from several other disease systems, not only from macaques, but also from humans. But to add to what Ted said, I think something that perhaps anyone designing those trials, if they are to occur, should take into account is what is going to happen in the real world setting. And I think one of the things that should be included as objectives of those studies would be what happens if they get given in the wrong order, what happens if you end up giving 2 doses of AD or 2 doses of MVA, or you end up giving MVA followed by AD. I mean, we would want to see some kind of information on what happens there, at least a lack of harm, because that's going to end up happening.
Thank you. Go this side.
Yeah, Yiming Shao from China CDC. Since this is a Grand Challenge meeting, we are facing a emergency crisis worldwide. I have 2 challenges. One is an easy one for GSK, and it's very good. You take up the task to promote the vaccine forward. My challenge is that with the Wellcome Trust money and Bill Gates Foundation money, you do that. Without the money, could you do that for the global crisis? Because I thought the Wellcome Trust and Foundation money can be used for the African country, maybe more. The second challenge is that I know all the vaccines and the drugs has been created mainly by NIH, by US government for anti-terrorist purpose. And I also know there's a problem of push that forward with the NIH alone, since the crises are, since the epidemic, small ones are in Africa for the last 30 years. So my challenge is to the global community to find a new mechanism to prepare all the product that goes to the end, not only for certain purpose, but for the global health as a whole. So all the vaccine or drugs are made like the VRC one, or 10 years ago. There should be long ago to finish all the clinical trials. So like during SARS, 10 years ago, during SARS, there also the way of doing global health business are not very good. So we have the next year, 2004, World Health Assembly building many new mechanisms And then we did very well for the avian flu response in the last 10 years. I think this is a time for us to, as a whole global village, to find a new mechanism to prepare for the future epidemics.
Thank you. David, do you want to respond to the first grand challenge?
Yes, well, the first question was about the GSK business case. And with this event, it's not financial. GSK has a vaccine that shows promise, and we want to bring it forward so that it can be used. The countries that need it most are some of the countries in the world least able to afford it. So I think this is a situation where partnership is called for, where it's needed, and many, many groups are stepping up. Not only the Wellcome Trust, not only the Bill and Melinda Gates Foundation, but the European Commission and the US government, obviously, and others are stepping forward to make this vaccine a reality. I would like to also comment on the priority of Ebola in the research list of priorities. I understand the WHO, and maybe Vasi could comment on this, has a list of 17 most neglected diseases. Ebola does not rank very high. In fact, Ebola is not even on the list. And that's because up until this year, it had been possible to stop outbreaks in villages with containment practices that were worked out in the Democratic Republic of the Congo and other places. And so prioritizing tuberculosis, malaria, HIV, and so on, some diseases like this have been prioritized lower. It would be wonderful to be able to work on all of them at the level they deserve.
Let me move on to in questions and invite our panelists maybe in their closing comment to think about the second challenge of how we as a global community rise to future emergent problems like this differently. But let's go here to this one.
Yes, Steve Cho from Princeton University. Recently, my laboratory developed a new technology that can enhance the existing assays sensitivity by 1 million times and reduce the assay incubation time by 100 times rather than hours, but less than a minute. So I would greatly appreciate that if you can introduce me to the companies who can need a high sensitivity and fast assay. And our technology is using the existing reagent. The advance is in the engineering side. So I think that would be very useful for early detection and fast detection. So I'll be grateful if you could give me some contacts.
Right. Maybe we can follow that up after the panel. But let me go to this question, then come back to Larry.
Thank you. Anna Berstein, Institute for Disease Modeling. So for almost 2 weeks, the media has been buzzing with claims that lamivudine, an HIV and hepatitis B drug, has efficacy against Ebola. And there are even rumors that some doctors have read this and are trying it. What is the plan to either debunk or confirm these claims?
I can start on that. So as you may know, researchers at USAMRIID have done, as well as NIAID, have done approved drug screens in vitro. This failed years ago and has recently been tested in the last week, and in 2 cell lines again showed no efficacy. So
that's right. That doesn't say that we shouldn't maybe explore that in an animal model, but at the moment, if you just look at cell-based assays, there doesn't seem to be a signal, and that's disappointing. I mean, the report was this Liberian doctor, or maybe it was Sierra Leone, gave this to 17 patients, and 15 of them survived, and that sounds like that was beneficial, but obviously, small numbers, don't know the circumstance.
Larry.
Yeah, Larry Corey from the Hutchinson. Couple comments. The first one, I personally applaud GSK for taking the risk and the liability of this, and I think you are to be commended. As someone who has lived firsthand the issue of the AD5 story as it relates to the STEP trial with the increased acquisition that went with that, I was pleased to see about the issue of doing this in a controlled way with the chimp ad. As much as we like to, think the positive, that one needs to be a little bit sanguine here. We've never been able to actually figure out why there was an increased acquisition. The leading hypothesis has been that it increased CD4 T cells because of cross-reaction. Chimp3 cross-reacts with human adeno. We will recruit adeno-specific T cells. Now, Ebola doesn't use CD4 or any T cell receptor that we for sure know that increases as acquisition, and we do have the envelope in there. But it is something that I think I was happy to hear about, because I think this is a double bell-shaped curve for chimp adeno. And I also think it creates a complication of where I had thought efficacy would go, which would be in a step wedge design. And I'm not sure increased acquisition is going to be well shown, or be pretty hard to show in a classical step wedge design. And I think that's something that the field actually needs to think through, whether something like that actually would be demonstrated in that way. I myself have thought that step wedge would actually probably be the best way to do efficacy, but I think we need to think through that a little bit.
Thanks, Eric. Next question.
Randall Wader from St. George's University School of Medicine. There was a comment earlier about the budget cuts and how they've impacted NIH research over the past 10 years. So my question is whether we're aware of anybody who's actually tracking the cost of intervening now that this seems to have gotten ahead of us. And if anyone's going to run that economic analysis of if we had actually not cut back on looking for an effective vaccine over the past decade, how much that would have cost us compared to how much getting this under control is going to cost us, so that we can go back to policymakers, government leaders, and say, look, this is what we've done by cutting back on our basic research.
That's a great question, and an important analysis that should be done, and I think it would come out looking pretty frightening in terms of what we are now taking on, just in terms of economic costs. In just one country, the US, will probably spend $1 billion over the course of the next few months in terms of trying to do the outreach, setting up those thousands of beds, invoking the US military to do a lot of the work. And that's just one country. Consider all the costs that are going into this from around the world. It's going to be quite an enormous number compared to what it might have taken to push the vaccines and the therapeutics forward, so that we would be at this point with those already as tools that we have ready to take on an epidemic of this sort.
Let me just take the questions that are at the microphone if they're really fast.
David Anderson from the Burnett Institute. I wondered if there's any evidence on whether HIV infection impacts the mortality or acquisition, which may interact with the observations on lamivudine.
I don't know of any. Sure. I mean, the case fatality in HIV uninfected people is already very high, so it may be difficult to resolve.
Go here.
Thank you. I'm Wallace White with Stratos Product Development, and I was curious from all of you about lessons learned from this so far. Obviously, we're still early, but are there both things that, for the preparedness for future outbreaks, we would want to do differently, and things that we're trying right now that you'd want to implement more widely on other situations to fast track and such?
Let me suggest that I'll ask each of the panelists to make a 1 or 2 sentence closing comment. Maybe you can incorporate a response to that. Let me take the final question.
Yeah, one last question actually. The thing is, now there is a consensus effort that you are generating huge amount of data set, and these are actually would be very useful for all the scientists who wants to work in Ebola. I'm just wondering, will you be making this data open access or publicly available so anyone who wants to work on these data sets can easily access and work on it without carrying patenting and publication issues? Yeah.
Okay. Thank you. Do you want to comment on the data access?
I just think data access is something that we increasingly have to focus on from NIH's perspective. The answer is yes, the data should be accessible immediately, even before publication. Look what Pardis Sabeti did with all those sequences out of the Ebola virus long before they were in a publication. People could go and look at them. That's the right model.
Great.
And I can add for GSK that we do post all of our data, and it is possible to gain access to subject level data through an agreement with GSK.
Right. Maybe just a quick final comment from each of the panelists. Vasi, since it's after midnight in Geneva, we'll let you go first.
Thanks. Just on the last point first, access to the data is clearly important. I think another part of this is very prompt reporting of results. And I think there's going to be, there is an existing WHO position that all clinical trials, interventional clinical trials, must have a clinical trial registry site. And there already are clinical trial registry sites for the USA, the VRC CHIMPAD trial and the UK CHIMPAD trial. And we will follow up to ensure that those exist for all of these trials as they come through. With clinicaltrials.gov, there's an opportunity there with the results component of clinicaltrials.gov to have facilitated access to results summaries when they become available, and I think that's going to be important. Yeah. In terms of closing remarks about the issue of prioritization, I think this is a very important point. Right now, the focus clearly is and should be on doing everything we can to make interventions available to affect the current epidemic. Beyond that, we need to look at what the lessons are that can be learned clearly. And I think one inevitable consequence of this is some re-looking at prioritization for, I work in vaccine development, and we're going to need to look again, probably I would say sometime during 2015, at what the prioritization for vaccine development is. Something that strikes me is that there was a really powerful comment that was made in the September 4 to 5 meeting by an ethicist called Michael Selgalid, who's based in Australia. And he said that his main comment, before he could make any comment about what should happen going forward, was the terrible prior wrong that was demonstrated by the lack of available clinical trial data from, for example, vaccines, so which I really support. In a better world, we would have had clinical trial data already. And secondly, the technical feasibility does not, it remains to be seen, but if you look at the macaque data, the technical feasibility is not the highest compared to some of the other pathogens that we're dealing with. Having said that, we're trying to square the circle of trying to know ahead of time what the emergencies are going to be in the future, and that's essentially close to impossible. We can try and do that, and I suspect what's going to end up happening is that there will be some more effort on filoviruses going forward, and we will have to get the smartest minds together from WHO's perspective and look again at our priorities for this sort of situation going forward. Thank you.
Thanks, Vasi. Ted, maybe we'll start with you and we'll just come down the line.
Sure.
Brief.
I think my closing remark would be this. Everyone's eye is on the ball of Ebola at the moment in West Africa and elsewhere. But it's not the only ball in play in West Africa. There's malaria, HIV, there's a list. And as the health systems creak and as trust in health services decreases because of the limited response we're able to offer, there will be a very serious consequence for all of the other conditions, for which the solution is to have more robust health systems there in the first instance, and health systems that are resilient enough that they can give the kind of early warning that is necessary. So I think one of the big lessons learned is that we will find there's a horrible harvest to be reaped of other conditions during this period while we focus on Ebola.
Yep.
I would completely endorse that. I would also say that we've been here before with SARS, with MERS coronavirus, both affecting actually much wealthier countries, but I would completely agree with the China CDC colleague who stood up that we really need to reevaluate how we undertake prioritization of development of products to tackle major emerging infectious disease threats, but also the surveillance systems in place to deal with those. I'm sure if you look back, we have a whole bunch of now SARS and other coronavirus vaccine candidates which have been sitting on shelves. And we don't want to be in the same situation again in future where we're always behind the curve of the next emerging epidemic.
I think there's going to be a repeating theme here. What we have learned in the process of the last few months of this intense activity is that there are a lot of resources and a lot of capable organizations and individuals that can come to the table in the presence of this emergency. But it hasn't been easy, and there are certainly lessons to learn about how we could be better prepared for such future events, be it a SARS or be it the next really serious influenza, which is waiting out there for us, and we can't be in any way confident that that's not going to happen in the relatively near future. So when we get through the emergent feeling that we're in the middle of right now, to try to back away from those specifics and ask across all of the global partners that are necessary to take on an emerging infectious disease. Do we really have those systems optimized, or will we go through a somewhat clunky procedure to try to get ourselves together the next time this comes around? I would hope we could learn from this and be much better oiled machine the next time.
David?
My comments are going to be from a vaccine perspective only. I think the lessons we need to learn are ahead of us. I've been just so impressed since the 8 August announcement from the WHO of Ebola as a global threat to how much we've done to start a phase 1 trial at the NIH on the 2nd of September. So, the leadership from the VRC and the WHO and many, many others, I've been most impressed. I checked my email at lunch. There was an email from our regulatory group. There were 3 pages of risk going into the phase 3 trial. Risk, suggested mitigation, risk, suggested mitigation. We're going to learn, unfortunately, we're going to learn a lot through this experience.
Chris?
Little to do, but to echo what's been said, I would say that it seems to me that both for this and for future similar situations, the fundamental barriers are not technical, actually. They're systems, they're resourcing. I think the technical hurdles for something like this are not as high as we think.
Yeah, I guess my concluding comment would be similar, but also expanding it. I think we're going to learn things in the face of this epidemic that will not just prepare us for the next one, but we're learning things about collaboration and partnership, about how to fast track things, how to get things into clinical trials more quickly, that we could be doing with things that are not, quote, emergencies. There's 4,000 deaths from Ebola. There's 600,000 deaths from malaria every year. How do we take some of these lessons about partnership, collaboration, data access, sharing, and apply them to the biggest killers of people in the world today? And with that, let me thank our panelists and thank the audience for being with us. Thank you.